Again, talking about aromatic amines, how they cause cancer. Again, as we saw yesterday with the polycyclic aromatic hydrocarbons, some cause cancer and some don't. And it takes organic chemistry to explain, in fact, why aromatic amines have this same difference. If the para position is open, and by that I simply mean a hydrogen para to the nitrogen, that's all that means, then in fact the compound will not cause cancer. If that position is blocked, either with an alkyl group here or as part of another ring, then in fact that compound is carcinogenic. And we'll look at the reason for that. And again, this goes back to England, back in the mid-50s. Some guy was working as a doctor in the dye industry. He began to look at death certificates and noticed a very high number of workers dying from bladder cancer over a long period of time, more than 20 years. It took them a long time to realize this, in fact. This compound, block para position, you can see causes a high incidence of bladder cancer. As it turns out, bladder cancer normally in the general population, the same control group, very slight risk. 11% of the workers got bladder cancer. With this compound, block para position, it's part of a ring, the other benzene ring, of course. Every single man that distilled that compound got bladder cancer. Not a big group, but all 15 men, 100% incidence of bladder cancer if the exposure is high enough. Compound is also present in cigarette smoke. Here's bladder cancer. You can see the tumor here and here. Very difficult to detect unless you have blood in the urine, because you're not going to feel pain. So this kind of summarizes Dr. Case's work. General male population, low percentage. Benzidine, which again is a block para position compound used to detect blood, actually. 12% of the workers got bladder cancer. Mixed amine synthesis. Those who made beta-naphthylamine, half of the group, and this is a very large group, more than 100, as you can see, got bladder cancer. Extremely indicative. And the workers that distilled the compound also got bladder cancer, 100%. Here's another slide that sort of summarizes that. I'm not going to spend a lot of time here. You can read about this. Workers that work with these compounds have a higher incidence. Truck drivers, I'll talk about later. Taxi drivers, for a different reason, have an incidence of bladder cancer that's more than you or I. Here's a compound, a simple aromatic amine, but the para position is blocked. It has a chlorine there. So what this means is, we're going to see in just a minute, metabolism then occurs on the nitrogen. And if that happens, that is, well, initiates the cascade into cancer, as you'll see. And you can see incidence was really very high in this plant, which I think is in Connecticut. It turns out if you burn meat, we talked about this yesterday, burn protein, glutamic acid, you produce aromatic amines. Now, fortunately, the bad one is the minor component compared to 1-aminonaphthalene, which is not a carcinogen. This compound was used to treat bladder cancer, Hodgkin's disease in Denmark, but the patients, even though they got better from Hodgkin's, which is cancer of the lymphatic system, got bladder cancer. Because in fact, this is metabolized. This nitrogen mustard, something like mustard gas we talked about, is metabolized down to the 2-aminonaphthalene, which is a carcinogen. Rat catchers used this to kill rats, thinking they were okay because here the nitrogen's at the 1 position, and this compound is hydrolyzed to the amine, but it turns out this is contaminated with a little bit of the 2-amino derivative. And so the rats had the last laugh here. The rat catchers got bladder cancer. These compounds are in tobacco smoke. All of the compounds in black, you can see, have an open para position, and they're not carcinogenic, but the ones in red are. And we're going to talk a lot about these two as well. Okay, this slide's important. The mode of action. Now, if the para position is blocked, you ingest this compound, hopefully not for real. Your body wants to make it into a water-soluble metabolite, so it'll go out in the urine. And the idea is to hydroxylate it, adding oxygen to all of these compounds, as we saw yesterday, makes it potentially more water-soluble. So hydroxylation occurs on the nitrogen. Now, this is an amide, actually, and this was about to be introduced in England as a new pesticide Then at the very last minute, it's back in the 50s or so, 60s, they decide maybe we should test it to see if it's carcinogenic. And it really is. Block para position, as we know now. So after hydroxylation, the OH is not a good leaving group, as you guys all know. So what your body does, in fact, is to make it into a better leaving group, the sulfate ester. The salt of a strong acid, sulfate, sulfuric acid, is a good leaving group. So your body converts it to the sulfate ester, and also the acetate ester is used sometimes. That's now a very good leaving group. And in fact, it can leave by a mechanism you'll see in a bit, where a piece of a protein can attack at this position. We're not going to talk about this chemistry. This is something we get into in Chem 63. But this appears to be the normal metabolism. Connection to a sulfur amino acid, and this is now excreted. But this compound also reacts with nucleic acid to give this product, and this is the product of aromatic electrophilic substitution on the 5-membered ring of guanosine. I'll show you that mechanism next. But in addition, the NH2 down here can also react with the nitrogen. And you might imagine SN2 displacement of sulfate, although the mechanism, as you'll see, is SN1. So this shows electrophilic attack on the nitritium ion, and this is the actual intermediate I'm going to show you in just a bit. And this then forms a covalent bond to the DNA. Anytime you have a covalent bond between an electrophile and DNA, you're in big trouble. It's very difficult to correct using repair enzymes. If you take this compound, block para position, you can make this in the laboratory. This is now the acetate ester of the N-hydroxylamine. Feed it to dogs or in the test tube, in fact, and you can isolate these adducts. Electrophilic substitution at that position gives that product, and then nucleophilic attack by the nitrogen directly on this nitrogen. And again, this causes bladder cancer in dogs. Okay, this slide's important. For a long time, it was thought that the mechanism was SN2 displacement, as I just said. Direct attack by a nucleophile on this nitrogen to give sulfate ester. You can imagine SN2 product here. And this would be what's called an SN2 prime attack, where you attack this benzene ring, shift the double bond. We're not going to talk too much about that. But now we know that the mechanism is SN1. You lose the sulfate, a tremendous leaving group, better than iodide probably, comparable to triflate maybe, or tosylate actually, you guys have had that, to give the nitrogen counterpart of a carbocation. And you haven't seen this before. This is called a nitritium ion. It's more reactive than a carbocation, and the reason for that is nitrogen is more electronegative than carbon, so putting a naked positive charge on nitrogen is really tough. It's a very reactive species, and this is what is formed in the nucleus of the cell, reacts with nucleophiles to give all of these products. Now you can write resonance forms and put that positive charge around the ring, and as you'll see, resonance theory again dictates the products. You can have a positive charge here, here, over here, at every other carbon, and in fact that's where the nucleophile attacks, as you guys would predict, and we'll see that a little bit later. Now the evidence for this mechanism is this compound's not a carcinogen, because now oxidation, metabolism occurs in the ring at the para position. We saw the first week of class, aminobenzene directs electrophilic attack ortho and para very rapidly, of course. You can't stop bromination after monobromination. And so that's what happens here. And this then is rapidly excreted, not a carcinogen. But if you block that position with either a fluorine or a methyl, now you direct metabolism to the nitrogen and it becomes carcinogenic. Again, block para position here and here, compounds in red, carcinogenic. Now this one has a block para position, but why isn't that carcinogenic? Well, it's already metabolized in the ring. And so this is already polar enough to be connected probably to a sugar molecule, a glucose, makes it soluble in the urine. So to summarize now, these guys, open para position, rapid oxidation, para to the nitrogen, maybe by way of the epoxide, we don't know for sure, and it goes out in the urine. We know that if you, in fact, synthesize this compound, this is what you would get if you metabolized it on nitrogen and feed that to dogs, it is carcinogenic, because now you have the pathway set up for formation of a sulfate ester and nitrinium ion. But fortunately, this doesn't happen if you're exposed to this compound. Then conversely, if you have the carcinogenic compound, oxidation is rapid on nitrogen, and then this initiates this cascade to cancer. Slower is metabolism in the ring, as it turns out, for whatever reason. Steric, perhaps, we just don't know, but this is the pathway if the para position is blocked. Okay, Azo dyes are related. Now these go way back. Azo dyes go way back. Butter yellow was used maybe by your great-grandmother to dye margarine to make it look more like butter. It's an azo dye that's yellow. This is carcinogenic because, or both of these are, because in fact the amine here blocked para position. The dimethyl compound is interesting because that's metabolized by loss of a methyl. And then you have NH, and once you have NH on the nitrogen, you get NOH and the sulfate ester. So these are carcinogenic, both of these, even though they don't have a proton initially on the nitrogen. This compound's already metabolized. This is not carcinogenic. And here, our bodies, in fact, can't deethylate a compound. This is a little bit beyond what you guys have to know. Again, we get into this in Chem 63. But a lot of these compounds were used by fishermen in the UK to dye maggots. Now, the maggots were frozen, and maggots are ugly white things, not appetizing to fish, so they're dyed red and blues and orange, and apparently that makes them more appetizing to fish. But they're frozen, they have to be thawed, and the fishermen would put the maggots dyed with these compounds in their mouth, and they develop bladder cancer. And also liver cancer. I wanted to show you liver cancer, and you can see all these yellow things are tumor. You can't operate on the liver to save it because of this. Now, butter yellow, again, was invented a long time ago. Napoleon actually didn't like white margarine, so he wanted it to look like butter, so they used butter yellow, this azo dye, which is a carcinogen, in fact, a long time ago. These are some of the compounds that were used. Now, the only bad one, well, these two are both carcinogenic. This one is not. But you can see the fishermen, lots of bladder cancer, and most of these guys were fishermen that used maggots that were dyed with azo compounds. Here are the maggots. You can see these are not appetizing. If you were a fish, you'd never eat that. But if you dye it, then you might. Okay, this is the mechanism now. With the dimethyl compound, you get initial demethylation, and that sets up the hydroxylation step. Now it can be hydroxylated on nitrogen. OH is not a good leaving group. It has to be made better, either the acetate or the sulfate. This is the acetate, and now this is a good leaving group. It leaves in an SN1 reaction to give a nitrinium ion, the nitrogen counterpart of a carbocation, a benzoic carbocation. The normal pathway, we think, is methionine and amino acid to do that. It certainly reacts with DNA. I'm not showing those products here. And also tyrosine. This is interesting. Tyrosine, of course, is the one amino acid with an activated benzene ring. It's in all of us. And that can act as a nucleophile to attack the positive charge that, by resonance, is at that position, as we saw just a little bit ago. Okay, the nitropolycyclic aromatic hydrocarbons are a relatively new group of compounds. They're found in diesel exhaust, diesel fuel exhaust. They're very carcinogenic because what happens, they're reduced in people to the corresponding amino compound. And if the nitro position in the nitroaromatic has a blocked para position, as these all do, it's predicted to be carcinogenic, and they are. as you can read about this. But also form when you burn various meats and wood products, as you can see, carcinogenic in lots of animals. And again, they're metabolized to the corresponding aromatic amine. Again, all the trucks on the interstates in this country are putting out soot that contain these nitro-PAHs. And it turns out truck drivers, of course, they don't go to the bathroom as often as they should. They're on the interstate driving for hours and hours, holding it in, giving more reaction time in the bladder for these compounds to initiate bladder cancer. Also taxi drivers. There's one compound, or actually a couple of compounds, that are produced naturally that have a nitro-PAH, blocked para position. These compounds were used as part of a diet regime in women. They're found in plants and herbs, and they cause kidney cancer for really the same reason. It gets reduced to an aromatic amine, blocked para position, and so a lot of women were coming down with cancer in a number of countries, as you can see. Okay, this slide really does illustrate the power of resonance that nature knows about, like you guys. So the nitro group is reduced in us by enzymes to give the amino compound. It's then oxidized, in this case on nitrogen, because the para position is blocked. It has to go someplace to make it water soluble. Your body doesn't realize what it's doing. Then you get a sulfate ester. Sulfate, again, the salt of sulfuric acid, a great leaving group. It leaves to give a nitrenium ion. And now by resonance, you can write the positive charge at a number of positions, here, 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 all the way around. Never at the wrong positions do you see attacked by the nucleophile. So, in fact, in this particular case, DNA attacks at this position. Now to give this product, just an SN2 type attack, or sorry, this is, direct attack here. That's the new bond. Or at this position, or electrophilic attack directly on the nitrogen, as predicted by resonance. Okay, any questions about aromatic amines at this point? We'll have time to talk about these later on the blackboard, if not tomorrow, later before the final. Okay, the third class of compounds, I alluded to this yesterday, are nitrosamines. These are responsible, we think, for the incidence of cancer of the esophagus in parts of China and in Africa as well. Once again, we have a bunch of compounds that are carcinogenic on the left and some that are not. We're just going to really talk about this one by and large, and also number 2 that's not carcinogenic. And you'll see organic chemistry that you've had answers the reason why you have the 2 lists of compounds. It's a very ugly disease, as you saw yesterday. This is cancer of the esophagus. Obviously, you can't take out the tumor. All of this lumpy tissue is cancer. This is in an animal that was exposed to butyl ethyl nitrosamine. Here's another compound. This is methyl benzyl nitrosamine, and this is an animal given a single dose of 246 parts per million. This is after about 270 days. And you can see all cancer. This kind of illustrates how these things go. You'll get more of this in med school, of course, but the beginning of a tumor here, then it breaks through the wall of the esophagus and then can spread. Tumor. It causes brain cancer as well. This is a nitrosourea, a different kind of compound. The mechanism is a little bit different. This is now 5 parts per million per day, or per week rather, after about 500 days. And you can see this is the tumor, I think, here and here. And cancer of the rectum caused by a slightly different compound. Just a few more. I know you're going to med school, so you might enjoy a lot of these. Perhaps not colon cancer. dimethylhydrazine. Okay. Okay, the initial mechanism that was proposed for this was to involve diazomethane, your old friend for making methyl esters from carboxylic acids. Because it turns out if you take this compound, give it to rats, you can isolate 7-methylguanine. And it's coming, apparently, they thought, from diazomethane. That is, you can write cleavage of this methyl group as formaldehyde. You'll see that in just a minute. That gives this compound. This can now dehydrate itself, lose H2O, and form diazomethane. That, again, you guys have used, if not in the last exam, maybe the coming exam, to make methyl esters. And they isolated this, and so it was widely assumed that diazomethane is the carcinogen, at least from the dimethylnitrosamine. The diethylnitrosamine gives the N-ethyl compound. But then a guy did a very clever experiment at the Cancer Institute in Omaha, Nebraska, a guy called Lajinsky. And what he did, he fed the rats deuterated dimethylnitrosamine, expecting to get this compound, because you would get CD2N2. That would never pick up another deuterium. That would react with DNA and give you CD2H. But what he got instead was an intact CD3 group. So even though diazomethane itself is a carcinogen, it's not derived from this compound to cause cancer. It's another mechanism. So this slide's the most important one in this category. What goes on here is oxidation at the alpha carbon. The alpha carbon in this case is the one attached to the nitrogen. You get hydroxylation. And I'll draw this on the blackboard. This is actually a 6-membered ring, not unlike the decarboxylation of a beta-keto acid or ester that we've had. So loss of formaldehyde gives this compound. Now this is a very good SN2 nucleophile, and we think that this is the proximate carcinogen, meaning it's the stuff that reacts with your DNA to methylate the DNA, as you saw in the previous slide. I would ignore this part. You don't really have to worry too much about this part. So what this means is if after you lose one of the alkyl groups on your dialkyl nitrosamine, you get a primary alkyl group left over, that will be carcinogenic. On the other hand, if you take this compound, this is not a carcinogen. Here, the alpha carbon, and it has to have a proton, so it only undergoes hydroxylation on this side. You lose formaldehyde, that gives this species. And now you guys should recognize that as not an SN2 substrate. Why is that? Yeah, it's a T-butyl group. In fact, it's a very good SN1 substrate, because not only do you get the T-butyl carbocation, but you get nitrogen as a leaving group. So this compound does not cause cancer, because after this demethylation to give this compound, this simply falls apart before it can attack DNA. And this will eventually give the alcohol, or by losing a proton, isobutylene, as we've seen a bunch of times before. That will not undergo the SN2 reaction, as we've had a bunch of times in this course already. This compound's not carcinogenic. Here, the alpha carbons have no protons, so this is not metabolized that way, at least. This compound is probably a little bit carcinogenic, because after hydroxylation, you lose acetone, and then you have an isopropyl group that, of course, can undergo the SN2 reaction and the SN1 reaction. So this is not a very potent compound. This one, of course, if you hydroxylate here, what would you lose, by the way, if you hydroxylate there and break that? Benzaldehyde, if you push electrons. And now the benzyl carbocation is very stable, so that can undergo fragmentation before it acts as an electrophile to DNA. Okay, we're going to not cover this. This is a little bit different, where the alpha carbon's already hydroxylated. So in view of the time, we're going to skip this one. Okay, the relevance of this is that we eat a lot of aromatic amines and also aliphatic amines. It turns out nitrisation ideally occurs in your stomach. The pH in your tummy is exactly right for converting secondary amines into these nitroso compounds, as you can see here. Proline, a very common amino acid in everything we eat, is converted to this compound, and these cyclic compounds, this one and this one, also carcinogenic, because if you go through the procedure, the mechanism, hydroxylate on one side, break this bond, you have a CH2 on this side, and that is a good SN2 substrate. And so both of those cause cancer. It turns out nitrate is reduced in your saliva to nitrite. And nitrite is the nasty stuff that reacts with the nitrogen now to give the nitroso compounds. This one's not a carcinogen, but this one is, of course. Here's that reduction. And again, the pH that will initiate the reaction between nitrous acid and a secondary amine in your stomach is ideal. Vitamin C, very important if you have a bologna sandwich or have bacon with your eggs, have some vitamin C, have some orange juice, because we know that will detoxify the nitrite or the nitrous acid before it can react to give you a nitrosamine. Again, some other sources, soy sauce can promote the formation of these things, small amounts. The dose makes the poison, of course. Again, vitamin C will block this reaction to some extent. Okay, there are other kinds of compounds. Even though these compounds don't have NH, it turns out, again, your body has the ability to demethylate tertiary amines. And in so doing, you make an NH compound that can be nitrosated, and you get nitroso compounds. The abuse of betel nuts is really very popular in parts of the world. These are compounds that are nuts, fruits that contain this compound that undergoes methylation and then can lead to mouth cancer, in fact. Okay, this is really very interesting. In fact, parts of China, there's an area where the people have a very high risk of getting cancer of the esophagus. There's another region where the amounts of nitrites and nitrates in the drinking water and the food are much lower. They have a lower incidence of cancer of the esophagus. What was done in this experiment some decades ago, as you can see, the high-risk area were given vitamin C after each meal. Not very much, 100 milligrams. That's a very small amount. Normally, what you would take would be half a gram or a full gram. And the nitrosamine formation was reduced drastically by the vitamin C. Just some more evidence about vitamin C and nitroso compounds. Later on the blackboard, I'm going to show you how nitrous acid works and how that's able to form the nitroso compound. I don't have a slide on that. There's a couple of compounds that I found, only two, that occur naturally, that are naturally occurring in nitroso compounds of secondary amines. And ironically, these are used in folk medicine, as you can see, but probably are carcinogenic. This is a case going back to Omaha. This is a lab technician working in the lab, was jilted by his girlfriend, decided to kill her by dumping dimethylnitrosamine in the lemonade in their house where she lived. Turns out she didn't like lemonade, but it killed two other people in the house. She didn't drink any. And later on, the autopsy revealed, in fact, that as I said earlier, you get methylation of the guanosine. Again, guanosine is the most nucleophilic part of DNA, and it is reacting with the dimethylnitrosamine. Another source of nitrosic compounds might be due to these nitrite esters. Now, this is used as a muscle relaxant, I guess. And what can happen now, if you have a secondary amine, it can react directly on the nitrogen, displacing the alcohol group after it picks up a proton and giving the N-nitrosic compound. And so it's been demonstrated you can take a secondary amine with this nitrite ester, it's called, and form the nitrosic compound. And again, we talked about this yesterday, this sarcoma. Really can be very serious, obviously. Okay, the next group of compounds, again, relatively new. These are compounds formed when you burn meat. So we have the aromatic amines, we have the polycyclic aromatic hydrocarbons, the heterocyclic amines. Now, tryptamine and tryptophan, these are amino acids that are in everything we eat. If you burn meat, they're actually produced, these compounds are produced from the tryptophan and also from histidine, in this case. Very carcinogenic in animals and produced when you burn any of these foods. As you can see. Okay, the mechanism again is exactly the same. Now, all of these compounds have a block para position, and they all have an NH2 group. And so conditions are ripe for forming the nitrinium ions. As you can see, oxidation occurs on the NH2 group. The hydroxylamine is converted to a better leaving group, in this case, the acetate ester. That leaves as acetate, which is a good leaving group, about like chloride, not quite as good. Nitrinium ion then can react, and again, using resonance, you can predict where it would react in the other positions. Not at these positions, because you have to have a proton to leave after you have attack. So again, you see attack directly on the nitrogen, electrophilic substitution here, nucleophilic attack by the NH2 right here, because the positive charge can be delocalized to that position, and of course already on the nitrogen as well. So again, these compounds are produced. It turns out beer and green tea extracts actually block the formation of these compounds. We don't know how beer does it yet. Green tea, we think it's the polyphenols that you read about actually have the ability to detoxify these particular metabolites. Any questions about any of these? Okay, the last group, apart from the natural compounds, are direct acting electrophiles. Again, as I said yesterday, DNA is a supernucleophile, so you have to be aware of its reacting with electrophiles. And you guys all know that these things, especially these two, are very good SN2 substrates. And in fact, they are carcinogenic. They don't need an enzyme to be active. They direct SN2 attack. A single dose of methyl iodide, 50 parts per million, will give massive sarcomas in animals in the drinking water usually, well below the lethal dose. So any compound that's a good electrophile, and we've talked about mustard gas before, aziridines, a three-membered ring, epoxides are good electrophiles in SN2 attack. Sulfate esters, as you can see over here, sulfates, all these guys are very deadly compounds. And you can see the tumors. This is dimethyl sulfate in a rat, and it didn't affect the vital organs, so the rat survives for a very long time, I think more than a year, with maybe 150 parts per million, I think. Here's another case, and this is interesting because this is an isopropyl group, and this clearly shows that a secondary alkyl group does undergo an SN2 attack on the DNA to give this tumor in a different kind of electrophile. Okay, mustard gas we've talked about before. I'm going to skip over these slides. Okay, to summarize, this slide you don't have to know. It's a little complicated, but these are all of the sites on one nucleic acid base, deoxyguanosine, that is the most nucleophilic of all the parts of DNA. You can get attack on this nitrogen, as we saw with dimethylnitrosamine, methyl iodide probably attacks here. Electrophilic attack, analogous to the reaction we had the first week of class on benzene, and just nucleophilic attack here. And also, I didn't mention this, but you also can see attack on the amide type oxygen as well. Okay, nature makes a lot of carcinogens, and the ultimate is aflatoxin, and this is responsible for a lot of the cancer in parts of Africa. Farmers who grow peanuts, basically, and also in parts of China. It's produced by a fungus, an Aspergillus fungus that grows on peanuts. It also grows on corn, in fact, and produces aflatoxins. There are 4 of these. The ones in red are much more carcinogenic than the ones on the right. Why is that, do you think? What's going on here, would you guess? Well, the only difference is the presence of a double bond. So what do you think happens with that double bond that tends to be activated by the oxygen next to it? It gets epoxidized. So it's like the epoxydiol we talked about yesterday. You form an epoxide here, and then that becomes very reactive in attacking DNA. And you can see 15 parts per billion gives 100% incidence in rats. Extremely carcinogenic. There's a city in China that is exposed to aflatoxin through the foods, corn probably, and also maybe peanuts. And you can see very high incidence of liver cancer. Also in Africa. This was in the news not that long ago in this country when it infected dog food and cat food, pet food. And here's what it looks like. So if you encountered corn that looks like this, be sure you don't buy it. Certainly don't eat it. So the mechanism is epoxidation, and now you have very rapid attack. This epoxide is even more reactive because it's on a 5-membered ring, more reactive than the epoxydiols we saw yesterday. So it undergoes attack by DNA, and like all epoxides, you get a trans product. And the nucleophilic attack occurs here. This is activated by the oxygen. I'm not going to get into that. Very toxic. All of these animals and humans as well. Again, a difference again between the rat and the mouse that we mentioned yesterday a little bit. You see, India's had some major problems as well. I'm going to skip over this one. This is, you'll certainly get that when you get to medical school as to what happens to the DNA when it is damaged. I don't want to get into that. It does have to do with the mutation, the point mutation we talked a little bit about yesterday, but skip over that. And skip over this. Asbestos, I mentioned this yesterday. Parts of California, Montana, the Libby mine in Montana and Idaho, producing asbestos. It causes lung cancer, but the major problem is cancer of the chest lining, mesothelioma, and you read about this or see this on TV quite a bit. You see workers, very high cancer deaths from a mesothelioma, this very rare tumor otherwise. And here are these fibers in the lung, and we think it's just a direct irritation. It's not a chemical transformation to some kind of metabolite, but just a direct irritation by these very fibers that have a certain shape and size. And you can see it's really quite severe. This occurred in New Mexico. The Native Americans there were selling silver, and they would use an asbestos board as an insulating material. To clean the board, they would pound it, releasing billions of fibers. And these guys came down with mesothelioma, and they also used old pipe insulation. In fact, only 5 cases, but population of only 2,000. In fact, all of these cases were Native Americans who were actually using the silver boards. I mentioned California, and this came to light not that long ago when a guy was planting a lawn in his front yard and uncovered this giant tube of natural asbestos, 35 feet long at about this diameter, and he had to abandon the house. In Turkey, a major problem, and this is now just coming to light, in fact, where half of all the people who died in these 2 villages died from mesothelioma. Not from asbestos per se, but a related fiber, erionite, that was used to build houses there. And you can see how many people died in these 2 villages from this very rare kind of cancer. It was also uncovered not that long ago in North Dakota, in fact, and this mineral was used, still maybe, used to pave the roads in this part of North Carolina. Right here, in fact. Okay, coffee. There was a big concern some time ago that caffeine coffee caused cancer of the pancreas, and this was a major concern, as you might imagine. Those studies were now shown not to be correct. We know that caffeine is not a carcinogen per se. Coffee beans aren't. Roasted coffee beans, of course, because you're heating things, you can produce a lot of the compounds we've been talking about. But there are lots of benefits of caffeine as well. You have to worry a little bit about these compounds we've talked about, but certainly it can be beneficial. But a lot of things have caffeine, as you may know. I mean, some of these just have a huge amount, and of course, if you drink too much of these, you can actually die. There are cases on record. These guys are loaded with caffeine, obviously. And here's some cases you can read about this of fatalities, too much caffeine, suicides, or mistakes, as you can read. But yeah, this guy is actually playing chess right now in the Middle East. He's our #1 player in the U.S. At every game, he'll have a Red Bull right next to him. As you can see, all of these are different matches. Way too much, of course. And again, Gary Larson. Okay, it turns out colon cancer is a major problem, men and women, as we saw yesterday. And some of the compounds we think might be involved are derived from fats. We talked about fat and obesity. And these are compounds isolated from feces. They're produced in your colon by bacteria. And they're mutagenic, and they might be the cause of colon cancer by forming a radical that's now, by resonance, very stable. And that's really all I know about this particular one. If you work over at the hop in the wood shop, the workshop, you want to make sure you wear a mask. Certain hardwoods in the dust are carcinogenic. Beech, mahogany, these woods, and we don't really know the cause. Is it like asbestos, a direct irritation, or are there chemicals in these dusts? But this came to light some years ago in Sweden, I think, or in England, I guess. Very high incidence of a rare cancer of the nasal passage. The tumor would start and then grow back into the brain. Very difficult to stop. Okay, we actually saw this before. I'm going to skip this over, how dichloromethane is metabolized to this unstable acid chloride that, again, can't be used to make benzaldehyde by the Friedel-Crafts acylation. Bad reaction number 1, as you may know. The chloroform is a little bit different. Chloroform is actually metabolized by hydroxylation, loss of hydrogen chloride, and that gives the war gas phosgene, which is a tremendous electrophile. It's a double acid chloride, can react with proteins now, and probably is carcinogenic if the dose is high enough. Okay, finally, I wanted to mention acrylamide. This has been in the news a lot recently, and it was thought that because it's formed when you make potato chips and crackers, it's produced during the frying process, it would be a carcinogen, and as you guys would recognize, it's a good Michael acceptor, alpha beta unsaturated carbonyl compound. But we think now it's probably not a carcinogen at the doses. If you eat a bag of potato chips, it's okay. And here is more evidence for that, and you can see. And I just wanted to close showing, I'm going to skip over this one, showing the mechanism. And the mechanism, even though it's a good Michael acceptor, it's thought that it's epoxidized first. And it's the epoxide now that can react with DNA at this position, now to give this compound. Ring opening of the epoxide would give this, as you can see. We know that acrolein, though, does undergo a Michael addition. That is here, alpha beta unsaturated aldehyde, nucleophilic attack here, and then you get the aminoaldehyde. We kind of had this on the last exam. Aminoaldehydes then would close to give an imine, or in this case, the carbanolamine. Okay, I'm going to skip over this. I wanted to get right to the end. Okay, these guys, epidemiologists, some time ago, looked at all the incidents of cancer in the US and tried to break it down by cause. And you can see there are lots of little causes. We've talked about some of these already. But the major causes, tobacco use, obviously, lung cancer, colon cancer, bladder cancer, all of these, cancer of the pancreas to some extent, and diet, too much fat, eating the wrong things. So the good news, I did want to close here, there are a lot of polyphenols that are in all vegetables and fruits and some beverages like tea and wine that are antioxidants. These guys destroy the carcinogens that we've been talking about. And you can read about it right here, and they're really very efficient, as you can see. Here's a compound found in chocolate and also wine and grapes. Very potent antioxidant. So some of the natural defenses, it turns out that the cells that are exposed to the environment in the nose, the trachea, the esophagus, they're changed all the time. So if they're damaged, they can be replaced and you'll be okay. We have repair enzymes I didn't talk about, but some of these have been identified that can go down along the damaged DNA and take out the bad nucleic acid base that's been modified. Glutathione I'll show you, and again, antioxidants. This is a compound that's been in the news quite a bit. Red wine, it's also in lots of fruits and vegetables. Resveratrol, and it's a very powerful antioxidant. You can read about this. Here are some compounds in green tea that have the same effect. Not found in black tea, as it turns out. Some of the benefits. Glutathione is our body's police molecule. Again, sulfur is a great nucleophile. This compound goes around in our bloodstream looking for electrophiles to kill. And it can destroy epoxides, nitrinium ions. All of these electrophiles that we talked about today and yesterday will react with glutathione. Okay, finally, these are some recommendations out of Japan. These are really very good. Balanced diet. You don't want to eat the same thing every day, even if it might appear to be beneficial. Not too much fat. Avoid fat. Too much alcohol, especially concentrated alcohol, that is 40% ethanol, is very corrosive, and that's what is in gin, vodka, and whiskey, unless it's diluted. Smoking, obviously. Vitamins, as we just saw today. and not just vitamin C, but vitamin A as well, and vitamin E to some extent. Fluids that are too hot, if you burn your tissue constantly, eventually it gives up and becomes a cancer. Salt is a promoter. It's not a carcinogen itself, but it seems to amplify the effects of carcinogens. Burned food, obviously. Fungal toxins, aflatoxin, for example. Sunlight, of course. Exercise. And cleanliness. And then one I added some years ago, breathe through your nose. Your nose is your natural filter to catch these particulates. You breathe through your mouth, unless you're a scuba diver, it goes right into your lungs.